DNA is a molecule that is made up of two separate strands. DNA is two strands that twist around each other in a helix-like structure. Each strand is built with four different chemical structures that are labeled A, C, G, and T, respectively, on the diagram. Whenever there is a T on one strand, the opposite strand will have an A. Whenever one strand has a C, the other strand will have a G. Each strand runs in opposite directions, which determines how DNA is replicated. The first bit of the replication process is when DNA unwinds from its helix.
The second step of replicating DNA is separating the two strands. The unzipping is carried out by an enzyme called helicase. After this process, the product leaves something called a replication fork. Each strand then affords a starting blueprint for creating the new strand of DNA.
The enzyme called primase then starts the next process. This enzyme created a small base of RNA called primer. This is the entry point for the new strand of DNA. An enzyme called DNA polymerase is then binded to the primer and will then make a new strand of DNA. The new strand is called the leading strand and is then built. The lagging strand is then made on the other side, on the other separated strand, going the opposite direction. The strands are made in a 5-3 direction.
On the other strand, the DNA polymerase can only form the strands in pieces called Okazaki fragments. Each Okazaki fragment is started off with an RNA primer. After that step is complete, DNA polymerase adds a short row of DNA bases in the 5' to 3' direction.
The next primer is then added further down on the lagging strand. Another Okazaki fragment is then formed and the process from before is then repeated. Once the new DNA has been created, the new enzyme called nexonuclease removes all the RNA primers from both strands of DNA. Gaps are left behind, so another DNA polymerase fills these gaps with DNA.
To complete the process, an enzyme called DNA ligase seals up all of the fragments of DNA in both strands to form a double strand. DNA replication is often labeled as semi-conservative, as the helix is made up of only one old and one new strand of DNA.